Good morning, everybody. The hour is 11 o'clock, and I see there's quite a few people who are in attendance. Welcome, everybody. We are kicking off our SASP virtual conferences, and we're glad that you're all here, and would invite you to future ones. We will keep you apprised of that. And today we have Dave Tabb as our guest speaker. Thank you for volunteering, Dave. And I'd just quickly like to introduce Dave for those who haven't met him. Dave got his PhD from the University of Washington in a discipline called molecular biotechnology, where he started out his interest in mass spectrometry. That was under the guidance of John R. Yates. And he finished his doctorate in 2003 and then went on to a postdoctoral position at the quite prestigious laboratory, national laboratory, Oak Ridge National Laboratory. And there he was in genomic analysis and systems modeling under Larimer. And there is where he started his interest in algorithms for database searching for spectra, and also worked on Fourier transforms for identifying peptides. So with that under his belt, he went on to become an assistant and then later associate professor at Vanderbilt, which was in biomedical informatics. And that was under Dan Masis, whom I actually met at one point. And he there worked on a lot of software. I'm not going to mention all of them, but they're all to do with mass spectrometry and using data from mass spectrometry efficiently. One is called ProteoWizard, which was still used extensively. Another one is ID Picker, and some of the work here sort of extends on that basis of the software that he developed there. Then in 2015, Dave arrived here in South Africa at the South African Tuberculosis Bioinformatics Initiative, and Dave and I have been working closely together since then, and have done our fair share of trying to enhance the bioinformatics education and bioinformatics research here in South Africa. So welcome, Dave, and we're looking forward to your talk, PCA-powered quality control for large-scale proteomics.
Thank you for that introduction, and I really appreciate the invitation to speak for SASP. This is a cool opportunity, so thank you very much. Today I want to talk about a subject that we frequently look past in biotechnology. If you were to run a sample through a genetic sequencer for RNA-seq or for whole genome sequencing, you might think that you'd get the same result every time. That's not very true. And when we get to the world of proteomics that relies on mass spectrometry, we find that the degree of run-to-run reproducibility that we see can frequently be quite problematic. So I wanted to spend a little time today talking about new research in the space of quality control that we've been expanding on since, gosh, I mean, my first paper on this was back at Vanderbilt, so I guess we're up to about 7 or 8 years of time with that now. So we'll start with the problems of technical complexity, how each of the different technologies that we staple together to make a bench platform contributes its own variability, and that variability tends to be additive, that variability that starts in an initial step may keep contributing onward. I am getting some weird sounds from my speaker, so I'm going to turn that right down. Okay, so then we'll talk a little bit about quality metrics and the challenges of how to interpret them. From there, we're going to talk about PCA, principal components analysis. And I'm going to try to introduce this topic so folks who end up using this for the first time are able to understand a little better what to expect from it. And then from there, we can talk about outliers, batch effects, and analysis of variance. And I'm really not sure why, but my speaker is just buzzing away. Careful, or I'll shut you off. All right, on we go. So let's talk about a typical set of technologies that we would use for shotgun proteomics. Ask what collection of proteins are found in this mixture. So we start with, as I noted, a protein mixture. Paradoxically, one of the first things we do makes that mixture much more complex by digesting it with something like trypsin to produce peptides. Those peptides, in turn, are hugely complex. Maybe you have tens of thousands, hundreds of thousands of different peptides all in this mixture. A lot of people will make use of something called fractionation in order to separate those peptides into different aliquots. And each of those aliquots can then be subjected to LC-MSMS, which is liquid chromatography, coupled to the mass spectrometer through electrospray ionization, which introduces this voltage that causes the peptide ions to take on protons and flow into the inlet of the mass spectrometer. And then we're going to have a couple different kinds of measurements for each of these ions. One will be a high resolution mass spectrum that asks, what is the accurate mass of this peptide? And the other is a tandem mass spectrum. So the tandem mass spectrum is not about all of the peptides in the mixture, but rather about the fragments produced when one particular peptide fragments apart. So you may have 10,000 copies of a particular peptide orbiting around in the mass spec, and it's through collision-induced dissociation, it's broken apart, and then we record the fragments in a tandem mass spectrum. These are the fundamental data for us, just as reads from a sequencer would be for RNA-seq or genome assembly teams. For us, tandem mass spectra are the inputs to our process of first database search to identify peptides, false discovery rate control in order to decide which spectra have been identified successfully, and finally inference of proteins that we've identified. There are, however, quite a lot of factors that can impact the quality of the data that we produce. So I'm not going to spend so much time talking about the downstream stuff about ID. That's a subject of another talk altogether. Instead, I'd like to talk about some of the different factors that can contribute variability upfront. So first off, a lot of times people are not doing whole cell lysates or dealing with thousands and thousands of proteins. Instead, they may be using something like co-immunoprecipitation if they want to see which proteins show up as partners to this one. Something like co-IP is very frequently used. They may be looking with particular interest at post-translational modifications. So they might use a titanium dioxide column to pull the phosphorylated peptides out of a mixture. These are examples of enrichment. Certainly, enrichment can have a tremendous effect on in contributing variability to the overall experiment. The use of an antibody twice against two aliquots of the same mixture is going to get somewhat different proteins back in complex. Certainly, people who make use of biofluids, such as serum or plasma, make quite a lot of use of depletion because these mixtures are because blood products are so heavily concentrated with just a very few major proteins. It becomes much harder to look at the interesting layer of proteins beneath them. So if we can pull those aside, that's great. And yet, the use of depletion columns will increase the variability that we see downstream. We generally have to denature proteins to cause them to go from their native folded states to be stretched out. And we're going to break the disulfide bonds through reduction and alkylation. And we need to digest those proteins up with trypsin. And much as we use trypsin every day, there are quite a few ways in which trypsin can behave variably depending on which buffers you're using, for example. Fractionation. There's a world of different techniques out there. Some of them are very efficient at separation and others are much less so. Liquid chromatography very frequently will show variations in the retention times at which peptides will show up. Electrospray has its own challenges. It could be that you get some variations in voltage. There could be an interruption in the flow from the column. You might have an unstable voltage source. There are lots of things that can go wrong with that process. How recently was the instrument tuned? What mixture was used for tuning it? How recently was the mass calibration tested for the instrument? How much crap has accumulated in the ion source of the mass spec? Inside the mass spec, there are all kinds of different electrodes used for transmitting packets of ions throughout the instrument. We'll talk about that in a minute. maybe the vacuum pump has changed in its efficiency over time, and maybe just through the process of aging. If this instrument is in its 3rd or 4th year of life, it may well be that the detector used for producing the signal has become corroded or has lost some of its ability to measure ion current effectively. Up at the right, we see that some of these effects all combine together under major headings, and then those major headings combine to create the overall variability. This is called an Ishikawa diagram or fishbone diagram. It's one that shows up very frequently in the quality control literature. Okay, so I think that we think of mass spectrometers as these pretty white boxes or beige boxes that sit in the sit on a bench top in the laboratory, and there's always a fair bit of noise around them because the vacuum pumps are running. You got to keep them pumped down for them to be useful. So I thought it would be useful to kind of show the path of ions as they move into, in this case, a Q-exactive instrument. So over here we have the source that's allowing ions to flow into the inlet here. And there's a capillary inside, often a heated capillary, that the ions move through. They reach this S lens. Now, everything shown in red here is an example that is altered as part of tuning. So if you tune, you may find that the before and after picture differs quite a lot. These other pieces tend to get examined in the case of an overall calibration. So we have a flatipole beam guide. Here we have the quadrupole that is used to decide which ions are allowed to pass into the instrument. And then the C-trap becomes sort of this, this intersection in the mass spec. The ions may move from the quadrupole here into the HCD cell for fragmentation, or if you're collecting in mass spectrum, the ions may flow through the quadrupole into the C-trap and then get conducted into the orbitrap. So you might think, if you're looking at an orbitrap instrument, gosh, that's really big. It's, got more depth than I can put my arms around. It's a huge box. The mass spec inside it, the mass analyzer itself, the Q-exactive, is very, very small by comparison. So there's an awful lot of ground to travel, and all of this space can get cruddy as ions that you don't really want in your instrument are accreting to the instrument itself. So it has to be cleaned from time to time. Obviously, when you're cleaning the insides of the instrument, your ability to keep it under vacuum is reduced. So you allow the machine to come up to atmospheric pressure, and then you need to do you need to drop its pressure in and bake it out. So all of these processes, even the ones for maintenance, potentially can cause variation. Now, the standard way that we use tandem mass spectrometers to identify proteins is this the data-dependent acquisition, DDA experiment. So I wanted to try to explain that for folks who might be relatively new to proteomics. Here we're looking at a mass spectrum. Remember I said we have two different kinds of measurements being produced for peptides. One of them in intact form, like in an MS, and another in tandem mass spectrometry. So we're here in these ions, in these tandem mass spectra at the right, we are looking at the fragments produced when a peptide ion was blown apart. But how does the instrument decide which ions should get blown apart? For that, we have a competing effect. The instrument wants to produce tandem mass spectra of everything. We can't really do that, though, because a lot of the ions that we would try to capture are not currently at a very high level. I mean, you can see tiny little bits down here in the grass like 708.35, a tiny little ion down here. It could be that there's enough of that ion to produce a meaningful tandem mass spectrum, or it could be that that ion is going to grow in intensity at the next MS scan. So it's hard to know at which moment is best to capture a tandem mass spectrum. So in effect, what happens is that the instrument says, I'm going to remember for the last 30 seconds which mass to charge I've produced a tandem mass spectrum from. And if a peak reappears at a location where it appeared in another mass spectrum in the last 30 seconds, the instrument says, hold on a second, I've seen you before. So we see that in a spectrum such as this, some of these big ions that look like obvious candidates for doing a data-dependent acquisition for producing a tandem mass spectrum are ignored by the instrument because it has already captured a tandem mass spectrum at that point. So essentially, once it can rule out all these different zones where it has already acquired a tandem mass spectrum, it can choose the most intense ions of what's left for producing tandem mass spectra. So that might seem very, very complete, that we're getting a great sampling of anything that shows its head in a mass spectrum. But the reality is that a great many of these peaks that appear in a mass spectrum are there and gone. There might be very, very little of them, and so they only appear for sampling in a single mass spectrum. If the instrument fails to select it at that time, that peptide is lost to us. So it may be that we're getting a reasonably good capture of tandem mass spectra from the peptides because it's a very simple mixture, but the reality is almost every proteomic experiment has far more peptide ions that create a peak at some point than that we actually observe through tandem mass spectrometry. This creates kind of a stochastic difference among experiments. This time when you run this experiment, the, let us say, 606 ion down here is big enough to produce a tandem mass spectrum. But the next time, it doesn't show its head dominantly enough to be selected for tandem mass spectrometry. So it may be that even though you're running the same sample, you will get different results. And here we're looking at an example of that. This is an early paper I wrote back in the years I was working with CPTAC, a big proteomics organization from the National Cancer Institute. Here we are looking at different LTQ instruments. These are linear ion traps and Orbitrap class instruments. Most of these, I think, were Orbitrap Veloses. And we asked, having examined a yeast proteome once, we have observed, let us say, 100% of peptides from that first experiment. Then we can ask, how much information do we gain if we add a second experiment just looking at the same sample exactly the same way again? And we see that in looking at an experiment the second time, we saw about 50% more peptides on the LTQs and about 30% on the Orbitraps. As if we added more and more replicates shooting exactly the same sample again and again and again, we saw that over 6 shots on an LTQ, we were able to see, let us say, about 120% more peptides than we could see in a single shot. So you could just keep on shooting samples and hope to get, at some point, a comprehensive list of all peptides. That curve is flattening, but it's not one of those things that you can really run to completion. You always have a lot of experiments competing for instrument time. So if you were to look at an individual protein and ask how many tandem mass spectra were observed for peptides from this protein, how much variability do we typically have for these proteins? And in this case, we have the 3 LTQs shown up above. Here's a relatively well-evidenced protein with generally more than 20 tandem mass spectra identified to it every time. We see that replicate 6 had a relatively low sensitivity for it, capturing 18 spectra, while replicate 3 had a pretty good sensitivity, capturing 29 spectra. This is the amount of variability we see when there is no change in the sample. In each of these 6 replicates, exactly the same sample is getting shot. So this is a sampling technology. Necessarily, there is going to be some amount of variability that appears just by random chance. You might ask then, if we claim something is different via mass spectrometry experiments, can we rely on that difference being real? And this is actually a question of some difficulty. We wrote a paper in 2015. This was a very, very hard lift paper for me. Took a lot of time to get us to a place where we were really comfortable with our answer. Many people in proteomics make use of a technology called iTrack or isobaric labeling for determining whether a protein is different between multiple samples. And here you can see that we were able to reproduce almost all of the differences we claimed from this statistical framework in another experiment. This was a case where the same pair of samples was run lots and lots and lots of times before we could assess the degree of differentiation we were doing. You see these bars at the top that are in kind of orange. Those are differences found by this experiment that were not confirmed anywhere else. So generally speaking, I think we can say that so long as you're pretty careful in the statistical framework you use to decide which proteins are differential, the differences you claim are reproducible if you were to repeat that experiment. That was something a lot of people had a lot of doubts about as we began the CPTAC project. So I was really grateful that we could shed some light on this kind of problematic concern. we've heard far too many people say proteomics is just not reproducible. And this was evidence that in fact it is. But you do have to be quite careful in your statistics. So if we have this kind of robustness about the data, and yet the problems associated with a sampling technology, it seems like it would be really valuable if we could characterize if each of these components along the way to a mass spec data file are actually working as they should. So you have an auto sampler and pumps over here. You have a LC column. You have the electrospray ion source, and you have all kinds of things going on in the mass spectrometer. And then you have the whole problem of protein identification, which you might think, well, it's happening in the computer. How could variability creep in? But I would point out that if you use Sequest versus Mascot versus Tandem versus MSGF+, each of these is going to come up with a different list of peptides from the same raw file. So even bioinformatic reproducibility is not guaranteed. So what we would like then is to be able to compute some metrics that capture information about how each of these different aspects is taking place. And one of the very first papers on quality metrics in this area was by my friend Paul Rudnick. So in 2010, he published the set of metrics that we were using as part of this CPTAC study to characterize individual instrument report files, the raw data produced by mass spectrometry. My team got really interested in this problem a couple of years later as we tried to robustify, to improve the robustness of the NIST framework. We wanted to create some software that could produce quality metrics, but do so in a way that the metrics were more stable. Over time, this idea emerged into the Quameter ID-free software. And the ID-free software is an unusual one because it says, I don't want to wait for the relatively costly in time protein identification process to get my results back. I want to characterize the raw file the minute it comes off of the instrument. This gives us the ability to do faster decision making about it. So Quameter included about 40 metrics that separated into similar categories: the extracted ion chromatograms, that is to say the pattern of intensity over time for peptides, the retention times to characterize liquid chromatography performance, the mass spectrometry, and the tandem mass spectrometry that's conducted. Overall, we focused on an expectation that the data we were working with were not always going to fall into neat normal distributions. And we tried to focus on quartiles to say, what is the 25th percentile, the 50th percentile, and the 75th percentile to help us understand the distribution of these values rather than just giving means, for example. And anytime we talked about intensity, we focused on log ratios of peaks to flatten out some of these distances and make these metrics more useful broadly. So you might, you can see 4 of the metrics that we use down at the bottom, just as examples of how this works. If a peak is eluting from chromatography, does it have a very wide peak, a minute, or does it have a very narrow peak, 10 seconds? We think of these peaks as being around half a minute, but sometimes they violate that rule. Similarly, we could ask how much of the full experiment in time was used to get to 25% of the total signal seen, to 50% of the total signal seen, or to 75% of the total signal seen. That's the retention time TIC, or total ion current integration. We also have things like how many peaks appear in the mass spectra. And naturally, we want to know what is the fastest rate at which the instrument is collecting tandem mass spectra. All of these are useful ways for us to characterize the instrument. I'm giving some more examples here of the different metrics that we can compute from this. And you can see that at the bottom, I'm showing a timeline for an experiment. Let us say in this experiment, we have just 20 tandem mass spectra, just a toy example. So we have the first 5 collected in the first quartile, the second 5 collected in the second quartile, and the third 5 are found in the third, and the fourth 5 are found in quartile 4. They have the same number of MSMS events in each of these timeframes. But you see that when the rate of MSMS scans increases, the time of that quartile contracts because the density of MSMS scans within it is higher. So this is kind of the way that we try to capture this information on a full basis. These metrics were published, by the way, back in 2014 in this paper by Xia Wang. We'll come back to her in a bit. So I want to make the case here that simply looking at a huge pile of metrics that come back from these experiments is a very difficult thing to do. How do you determine which bits of information are most interesting in evaluating whether the instrument is giving you a stable result or not? So I would note that many of these measurements could very well be redundant. They may contain mutual information, or they may be correlated to each other. Now, that doesn't always mean that when X is high, Y is high. It can mean that when X is high, Y is low, and when X is low, Y is high. You can have these inverse correlations as well. In both cases, that's shared information between these channels. So principal components analysis, thing that came to our attention right away as an obvious way to do a multivariate analysis of these metrics that came back. So for this, I want to explain a little bit about how PCA works in a way that I hope will be accessible to everybody. So if you are a very advanced biostatistician, this is going to disappoint you a bit, I'm sorry. But if you're a relatively new graduate student, I hope that this can really help you. First off, it's called principal components analysis because it is starting with these measurements you've made and turning them into components. So you have n metrics. The Quameter software creates 40 metrics. So it accepts a table of these 40 metrics for a large number of samples. It then wants to translate these metrics into some sort of weighted sum. So I'm going to have a weight attached to the first metric and a weight attached to the second metric and a weight attached to the third. Each of those values is going to get multiplied by its weight, and then we're going to add it all together to call that a component. Now, importantly, PCA is going to do this combination in a way that attempts to maximize the amount of variability that can be a variance that can be explained in the data, so that the first principal component that comes back from PCA is the one that explains most of the variance in the data, or the largest amount of variance in the data. The second component that comes back explains the second most variance in the data, and so on. And these are intended to be non-correlated, that these different components, even though the input data may be correlated, the components that are created from it are not. I would also note that you'll frequently, I would say the principal components analysis very, very frequently suffers from misspelling. A lot of people are very convinced that principal, that they mean the principle of the thing. But I want to point out that PCA is ranking components by variability to show you the principal components for explaining the variability. So let's look at this in a graphical way. I think many people can get their heads wrapped around the idea that we have a cloud of dots in this space. So let us imagine that each of these dots represents an LC-MS-MS experiment. And here we're measuring just 3 metrics on each one. Maybe it's the number of MS scans, the number of MS-MS scans, and the fraction of MS-MS that were +2s, something like that. So this creates a 3-dimensional space in which we have this cloud of dots. Now, as you look at this cloud of dots, you can see that if you look at it from here, you don't have a huge amount of variance. But if you turn it sideways, this cloud kind of separates from left to right. And you can say, ah, that's the biggest variance I have in this set. And if it's kind of tall this way, then it might be that if we were to look from left to right that way, we would also see more variance. So we can do what's called a projection pursuit to rotate these data around, not changing their relationships to each other, just rotating the cloud to say which way does it appear longest, thus giving us the most variance across this study. Okay, then we can ask what is the cross to that, a right angle to that allows us to explain the second largest amount of variability. So here you can see that we started with 3 dimensions, but on the right we're only looking at it in 2 dimensions. So we have another dimension that we can work with here, but most typically we see that in PCA, very frequently, however many dimensions you start with, most people will visualize them on a principal component 1 versus principal component 2 axis. Now, this is a really widespread data set that you frequently see used in data science classes. Here, each dot represents a particular flower, and we're looking at sepal length and width and petal length and width, and so each of these points has a kind of a four value address in 4-dimensional space. Did I just talk about 4 dimensions? I just talked about 4 dimensions because the more metrics you have, the more dimensions this PCA is going to be working with. So we can't really visualize a 4D space very meaningfully on screen, or frankly, even if we had holograms, we would have some difficulty with that. But in this case, PCA has said, if we combine the values in this way, we find that principal component 1 explains the most variance in the data set, and principal component 2 explains the second most. So combining the 4 values that we had for each plant, we can now say, for example, that this plant falls right in the middle on principal component 2 and has a relatively high value on principal component 1. Okay, so that's great. We need to be able to keep enough components in our visualizations that we can account for substantial variability. That's kind of a difficult concept, so I'll just say that if your first principal component explains only 10% of all variance, and principal component 2 explains maybe only 6% of all variance, the two together are only going to show 16% of all variability in the data set, which means that even though this point seems to appear right in this neat little space right here, it might be that if we were to view the third dimension, we would see it lifting out from the screen towards us, right? So we're looking at a projection in 2 dimensions of data, in this case, that are 4-dimensional. And we always have to keep that in mind when we make a PCA plot from it. I want to also draw a connection here to unsupervised learning. PCA does not know the labels, the group associations of these files. So in this case, each of these dots represents a different plant, and they come from 3 different classes, 3 different species, Setosa, Virginica, and Versicolor. So each of these dots is going to fall into one of the 3 colors, green, yellow, or blue. But PCA does not receive that information. It doesn't see it at all. So PCA is not trying to find the best separation between classes. It's not capable of that. It is instead saying, where is the variance we see in the metrics here? And it may be that your 2 classes fall right on top of each other, or they might separate very neatly. Essentially, if the most dominant reason that some of these data points have high values and low values is which class they're from, then principal component 1 is likely to separate the 2 classes from each other. And that's brilliant, but PCA is not doing that as a way to optimize the separation. For that, you would really need something like support vector machines in a supervised learning technique. Okay, so we've spent a little time talking about quality metrics. We've spent a little bit of time talking about PCA. Let's talk about how this plays out in proteomics. My friend Wout Bittermje put together this nice paper back in 2017 that talks about some of the really valuable things that we can seek to do with quality metrics. Imagine that you're a journal editor and you're trying to decide whether a proteomics paper should be accepted or not. It might be valuable to you to know that the quality metrics show that this data set checks out or not. Right now, they don't use that information because they frequently don't have it available to them. What if you are running a core facility and you want to decide if Grumpy, Sleepy, and Doc, three mass spectrometers on one side of your office, that all of them are in fine shape, or maybe Doc is being a bit grumpy of his own accord? Well, then it may be that you need quality metrics in order to evaluate whether that instrument is up to scratch or not. I'm going to just talk about 5 roles that appear here at the right. It might be that project management is your role, and you're trying to decide, is this facility ready to run our uber critical samples that we've worked 2 years to collect? It might be that you're a bioinformaticist like me, and you're trying to discern whether these data can be reused or not. So you're looking at a repository and trying to decide if they're useful. Maybe I'm a reviewer for a paper and I want to evaluate the quality aspects of the data collection. It could be that I'm a technologist. I'm trying to improve the way I use my instruments. It might be that I'm an instrument operator and I'm just trying to say, what is broken about this machine? It's been vexing me so much. So there are lots of different questions that we might ask as proteomics users. So I'd like to start with this paper that introduced the Quameter ID-free paper back in 2014. I formed a collaboration with my friend Xia Wang up at University of Cincinnati. I'd spent a lot of time in the proteomics community. She spent a lot of time in the biostatistics community. So we worked together, learning each other's language, because frankly, bioinformaticists and biostatisticians and bench experimentalists do not always speak in a way that everybody understands each other. So we talked about Quameter as a way to translate each raw file into a vector of 40 numbers. These are the metrics that evaluate it. PCA is then able to discern from this batch of numbers that's been produced over a large number of files how these where the variance is to be found in these data sets. So I would start on the right with the problem of outliers. If you have 200 experiments, you've already invested probably more than 200 hours of instrument time in this experiment, and you may want to know, are we done yet? Do we have good data from all of these experiments? If you see a situation like the one at the right, you might be concerned that a couple of these files seem to drift away from the main body of experiments in PC space. So finding outliers could help you to point to experiments that you might want to redo if they're not reliable, or to excise them from the experiment in another case. At the left, we have batch effect recognition. I think that we're aware from the earlier part of this talk that if you simply run the same instrument on the same sample, the data will vary over time. But I want to point out that very frequently, people in proteomics fall into the trap of running their cohorts separately. And this is something that it's very hard to do anything to fix after the fact. So let us say I'm trying to put together a case control study for TB diagnosis. If I run all of my cases in June and only have only give my controls to them in August, you can see then that there's this period of months of time that's passed between when the first cohort was collected and when the second was collected. This can create a huge problem because now the biology of how these two samples are different from each other is competing with differences in how the instrument is feeling between this time and that time. And we found things as simple as, are they painting the hallway outside, can actually play a really big role in affecting the mass spectrometry data that we produce. You might think, well, gosh, I'm glad I don't use a mass spec. I'm a sequencing person. But the reality is sequencers also have good days and bad days. Batch effects are a real thing. It is essentially always a bad idea to run one cohort now and another cohort when the time arrives. If it is possible to put these together, or to run half of this batch and half of the other batch at this time point and so on at the later point, then maybe you could be okay. But generally speaking, if the time difference for the instrument is confounded with cohort differences, it's going to be very hard to come up with anything biologically meaningful from your data. And in some cases, we would like to be able to evaluate variability sources. What are the causes by which we have different results? A friend of mine recently sent some cell lysates to multiple proteomics facilities in South Africa to ask whether the data that came back were equally sensitive among these different facilities. So being able to discern not only is it higher or lower between these sites, but how reproducible were the values that came back from these data sets is important. Could it be that a technician with good hands used the instrument on this day and the trainee used it on the next day and isn't quite as experienced yet with how to make the instrument perform? It could be that the operator of the instrument can cause a difference in the data that come back. All right, now this is one of the largest proteome data sets that I've had the fun of working with personally. This was part of the CPTAC at National Cancer Institute colon cancer project. So we were evaluating 95 different samples, and each of those samples was not represented by just a quick 1-hour experiment on the instrument. Instead, we were using fractionation up front by something called basic reverse phase liquid chromatography. So each sample was represented by 15 raw files, not just one. As you can see, over 95 experiments and 15 fractions, you have a very, very large number of files. In total, we were, I believe the count was above 1,400 that we encountered for just this aspect of the data set, and there were others as well. So what we've done here is to use different colors and different letters to signify different periods within this. So very first experiments performed are shown in kind of that deep red, and all appear kind of at the upper right of this PCA diagram up here. The second set was run right on its heels, but maybe they'd done a cleaning or something, because the data all showed up down here. You can see that the latter parts of experiment set 1 are shown by these reddish letters in the black cloud. So the first set of samples, the second set of samples, both look quite different over the space of 8 months of running the same instrument the same way on the same kinds of samples. So here we see the first batch and continuing into this cloud, the third batch spreads out across this space, and then we get into the dark blue, the kind of navy blue ones. You can see that the first experiments from that batch show up here and the others appear down here. You might ask, gosh, is that meaningful? This is a very difficult thing to ask. So one of the things we asked about were, do the identification sensitivity we achieved in these experiments vary in response to principal components? You can read the whole paper if you like back in 2015. But what I want to note here is that if you had any doubts that mass spectrometry instrumentation varies in performance as a function of time, I hope that this will set those to rest. Of course they do, and it's not limited to mass spectrometry. Now, I'd like to talk a little bit about Marina Creek, who defended her PhD yesterday. Yeah, really proud of her for this one. So here we're looking at outliers. So it could be that if you're using principal components analysis, that you are using an approach that is really subject to getting distorted by just a few outliers in the data sets. So in this case, we're using something called robust PCA, which is relatively insensitive to a few experiments being very far from the main cloud. We are looking at 120 raw files that were produced by 4 gel CMS experiments. This is a fractionation by gel for 3 different tuberculosis strains. This was collected here in South Africa at Stellenbosch University. So what we saw was that in principal component space, again from robust PCA, these 2 points were quite a lot further from the main cloud than anything else. And we were kind of worried about this. When we began looking at this more closely, we suddenly realized that although these raw files were named in a way to suggest that they were all run together, that all 120 were basically run back to back, the reality was that the instrument operator noticed something after these 2 data points had been collected, these 2 raw files had been produced. And so they shut down the instrument and they began cleaning everything and fixing it up. And so it was about, I believe, 3 weeks that passed between the time that these 2 flawed experiments were run before she started running the next set of experiments. And those new experiments then fell right back in the cloud with everything else. So you might reasonably ask, why didn't she rerun these 2? The answer is it costs money to keep the instrument running. And in this case, those two files had not been detected as faulty by their day-to-day quality assurance steps. Okay, so this is an example that data sets that go to publication may yet contain quality outliers that are problematic. I would also note that not everyone is doing data-dependent acquisition or shotgun proteomics. There's a new technology, well, not that new at this point, but for the last 10 years or so, people have been working on tuning up technologies for data-independent acquisition. One type of this is called swath. So in this case, I'm showing a paper from 2012 on this subject. You can see that they are creating layered windows that all overlap with each other. So we see that a window from about, oh, sorry, let me back up here. That you have a window here running from about 450 m/z to 475 m/z. This is a swath window, and within that, all peptide ions that have a precursor that falls in that range get co-fragmented, and their fragments are all recorded in a single tandem mass spectrum. So that is rather different than normal shotgun proteomics, where we are fragmenting basically ions of one peptide at a given time. So what we see then is that these data-independent acquisition experiments have the potential to produce a fair amount of variability in their own right. So Marina Creek, again, has created a software called SWAMI that allows us to compute metrics that evaluate whether the technologies behind swath are producing data that are relatively uniform in density, or whether we have some swaths that are doing much less work than others. And helpfully, she's created some software called Assurance to make it a lot easier for people to use these tools. You might be thinking, I don't have any time to learn R, or gosh, learning Python is a pipe dream. That's just not going to happen. Well, it's very handy then to have software like Assurance to handle that for you. Now, I would point out that I'm a professor and I get really, really busy. And so my ability to sit down and knock out a custom R script for every project I collaborate in is pretty limited. So when I was asked by some friends of mine over at UCT to look at a data set they produced in conjunction with CPGR, I was really excited about it. So in this case, they had, oh gosh, I don't remember the exact number of files, but what I wanted to know was, for this pile of raw files, do we see any that look like they're just in a completely different space than everything else? So I was able to use a Quameter to generate a table of metrics from those files. We could then feed them to PCA and produce this image. So here, I didn't have to write up the PCA code myself. I could simply use Assurance to make that visualization for me. And it actually has quite a lot of nice extra features too. So if you wanted to use machine learning, you can say, having used having shot this sample on this instrument over a space of a couple months, I want to say, well, this one and this one and this one are clearly bad. The others look much better. So you can use machine learning here to separate, to train a classifier to say, this is what bad experiments look like. This is what good experiments look like. If you have a system like that, you can then shoot a new sample and say, well, here are the data that came back from that. Does the classifier put it in the good pile or the bad pile? Very valuable. I would be remiss if I didn't mention that for the last several years, I've been working with an organization, part of the Human Proteomics Organization, the Proteomics Standards Initiative, to create a new standard way for people to be able to communicate information from quality metrics generators to tools downstream that can visualize it, that can use it for outlier detection, that could help us produce a dashboard for all the instruments that are in operation in the lab, which ones are doing well, which ones are doing poorly. And we've taken things in a relatively new direction. HupoPSI is widely known for having created the MZML format for storing raw data from mass spectrometry in XML. We've opted to use an approach called JSON to create a very compact, very easy to parse stream of information to pass information from one tool to another. So just because some of these metrics fit into nice square tables that experiment 1, experiment 3, metric, we can instead create information that's got a complex structure and feed that to downstream tools via MZQC. So certainly, proteomics quality control software is growing in maturity over time. There are a lot of people working in this space and coming up with cool ideas for how to use this information. In the old days, I remember people in the mass spec core facility at Vanderbilt shooting BSA on the instrument and then kind of looking at the data manually to decide, well, that ion is visible, that ion is visible, the mass values all seem reasonable, the chromatograms look sensible, okay, on we go. But eyeballing it like that is not a really scalable option. We need to have software that can support those activities. Statistical models based on QC metrics can certainly recognize outliers. They're quite handy for looking for batch effects and certainly for understanding what factors are contributing variability to downstream data is just necessary, of course. PCA is ubiquitous. Certainly, we didn't create it in the space of proteomics. We're borrowing a technique that has shown up all over the place. So given that it's going to show up on research posters and presentations that you see for the rest of your career, I think it's very valuable for you to understand a little bit more of how it works. So I hope we were able to contribute that today.